morning. My name is Jasmin Gao.
I'm Rose Hong, and our project is on the effects of fibrin gel scaffolds and dexamethasone on the differentiation of human dental pulp stem cells for applications in regenerative endodontics.
Annually, over 16 million endodontic procedures are performed in the United States alone. Many of these procedures are for children, as according to Northview et al., traumatic dental injuries affect nearly 30% of children. Traditionally, infected dental pulp is removed and replaced with an inorganic material in a process known as root canal therapy. However, the dental pulp provides nutrition and detects potential pathogens, so removing it poses numerous complications. Furthermore, infected dental pulp in children presents a unique set of challenges, as children often have undeveloped roots. Therefore, it is crucial to stabilize and anchor the tooth using the vast amount of pulp present in children. A current treatment called pulp revascularization aims to restore the root canal and surrounding tissue for continued development. This process stimulates a blood clot by over-instrumentation, which then draws upon stem cells from the periapical region. The blood clot is able to stimulate regeneration through molecular signaling to the stem cells. However, this process is not only extremely invasive, but also has low success rates due to a difficulty in forming the blood clot and also the formation of undesired non-pulp-like tissues. Pulp regeneration relies upon the use of dental pulp stem cells, which are found in the pulp chamber of the tooth. DPSCs are a type of mesenchymal stem cell that is highly accessible, displays rapid proliferation rates, and can differentiate into odontoblastic cell lineages. Oftentimes, DPSCs are treated with dexamethasone in vitro, as dex is a glucocorticoid that is known to induce differentiation. However, previous studies have shown that dex may have adverse side effects in vivo, including immunosuppression, delayed wound healing, and suppressed calcium absorption.
Current scaffolds for differentiating dental pulp stem cells include polyglycolic acid, polylactic acid, and polyurethanes. However, these scaffolds are largely limited by concerns with biocompatibility, degradation rates, and toxic degradation products. Additionally, DPSCs failed to fill the pulp canal space when plated on collagen-based hydrogels because the cells cause dramatic contractions. Therefore, we attempted to use fibrin to facilitate the formation of pulp-like tissues. Fibrin is a product of the blood coagulation cascade that is formed when thrombin cleaves fibrinopeptides from fibrinogen, allowing for non-pole interactions that produce the network. Fibrin gels exhibit outstanding biocompatibility, promote cell adhesion, degrade controllably through plasma-mediated fibrinolysis, and release non-toxic degradation products. Additionally, thrombin's mechanism enables it to form tight gel networks in short clot times, and its stability of about 6 hours eliminates the need for additional heater chemicals to remove it, allowing fibrin's composition to remain relatively unchanged over time. These properties make fibrin a highly attractive biomaterial for tissue engineering. With this in mind, we simulated a blood clot in vitro to investigate the ability of fibrin gels to support the proliferation and differentiation of dental pulp stem cells and study the effects of DEX on the cells. In part 1 of our project, we formed our gels and identified them by their concentrations of fibrinogen and thrombin, such that a gel prepared with 4 mg per ml fibrinogen and 2 units per ml thrombin was named 4-2. We created 5 samples each of 4-2, 8-2, and 15-2 gels on P35 dishes. As you can see in Figure 5a, this was one of our initial attempts at creating a fibrin gel that resulted in thick fibers. But after multiple attempts with various techniques, we were able to create the homogeneous gels that we eventually plated cells on, as you can see in Figures 5b through d.
We put the gels in non-differentiation media composed of alpha MEM supplemented with 10% fetal bovine serum, 200 millimolar ascorbic acid, and 10 millimolar beta-glycerol phosphate, and penicillin streptomycin. After 30 minutes of incubation, we removed 1 gel of each concentration and measured its elastic modulus with a rheometer to compare the strengths of the gels. Afterwards, we used AV1 DPSCs previously isolated by our mentor and plated them on the remaining fibrin gels and also on tissue culture plastic as a control at a cell density of 2.5 times 10 to the third cells per centimeter squared. On day 2, we added differentiation media containing 10 nanomolar DEX to half of the samples as a positive control. On day 2 and day 4, we then imaged the cells using an optical microscope to observe cell motion and cell proliferation.
We conducted RT-PCR on the 4-2, 15-2, and TCP to see the relative expression levels of differentiation markers. RNA was isolated using the QIAGEN RNA-Z mini kit. It was then reverse transcribed into cDNA using superscript 2 reverse transcriptase, and the cDNA was used as a template for PCR using the CyberGreen PCR kit. We looked at 2 differentiation markers normalized with 18S. One was an early differentiation marker, alkaline phosphatase, or ALP, and the other was a late differentiation marker, osteocalcin, or OCN. We also looked at 3 genes normalized with glyceraldehyde 3-phosphate dehydrogenase, or GAPDH, and these are plasminogen activator, or UPA, plasminogen activator inhibitor, or PAI1, and plasminogen activator receptor, or UPAR. The forward and reverse primers for these are listed in Table 2. Our rheology tests show the strength of each gel under the force of a plate shearing down on the gel. As you can see in Figure 6, the elastic modulus versus shear stress curve is significantly higher for the 15-2 gel than for both the 8-2 and 4-2 gels, and the 8-2 is higher than the 4-2. Figure 7 demonstrates that our 15-2 gel was stiffer than both the 8-2 and 4-2 gels. As the fibrinogen concentration of the gel increased, the average elastic modulus of the gel increased as well. Figure 8 demonstrates that the maximum shear stress a gel was able to withstand before total deformation increased as the fibrinogen concentration of the gel increased as well, indicating that maximum shear stress was directly related to the fibrinogen concentration.
On day 2 and day 4, we imaged the cells and observed that the cells had extended and adhered well to the surface of the fibrin gels. As you can see, the cells appear to have a high cell plating efficiency across all 3 concentrations. You can see from the images that the cell numbers increased significantly from day 2 in Figure 9 to day 4 in Figure 10. Using image day, we then determined a cell count number for each of the gels, which we then used to calculate the DPSC doubling time. As you can see in Figure 11, the differences in DPSC doubling times were not statistically significant across the various concentrations and also across plus and minus Dex gels. This indicated that external factors did not significantly affect population doubling time and that the cells had similar proliferation rates.
On day 2, we noticed that the DPSCs formed circular and semicircular regions on the gels as they proliferated. Circular regions were much more prominent on minus Dex gels, as shown in Figure 12a, than on plus Dex gels, as shown in Figure 12b. Using image J, we compared the number of circular and semicircular regions on the gels. Although Figure 13 demonstrates that the difference in the number of semicircular regions was not statistically significant, Figure 14 demonstrates that circular regions occurred significantly more often on minus Dex gels than on plus Dex gels. This indicated that DPSCs were extremely sensitive to the presence of Dex because it interfered with their ability to sense the fibrin. On the other hand, as the minus Dex fibrin gels deformed, DPSCs were able to react to substrate mechanics and self-organize into curved regions.
ALP is an early differentiation marker of odontogenesis, and as you can see from Figure 15, ALP activity levels of cells cultured on the 15-2 minus Dex gel is significantly higher than that of cells cultured on the 15-2 plus Dex gel. This indicates that the cells are differentiating and that on a higher fibrinogen concentration, the minus Dex gel is able to enable upregulation of ALP. OCN plays an important role in dentin formation and is thus often used as a confirmation marker for odontogenesis. As you can see in Figure 16, the OCN levels of cells cultured on the 4-2 and 15-2 minus Dex gel is significantly higher than that on the TCP. You can also see that OCN levels are significantly higher on the minus Dex gels than on the plus Dex gels. This indicated that on fibrin gels, OCN was actually downregulated by dexamethasone. From this, we could also see that the fibrin gels are able to independently enable upregulation of OCN.
We noticed that significant degradation had occurred on the fibrin gels without Dex, while those with Dex, especially the harder 15-2 plus Dex sample, had hardly degraded. Plasmin is a well-known protease that degrades blood clots and is produced when plasmin activator cleaves plasminogen. We therefore decided to explore the effects of Dex on plasminogen activation. We checked for the gene expression levels of UPA, PAI1, and UPAR, and found that adding Dex inhibited the production of plasminogen activator. As you can see in Figure 19 left, no significant difference in UPA production was noted with increasing gel modulus in the absence of Dex, but a significant decrease with increasing modulus was noted in the presence of Dex. Therefore, Dex inhibited UPA and acted synergistically with the fibrin mechanical response to increase inhibition factor as the gel modulus increased. This helped explain the lack of fibrinolysis on the 15-2 plus Dex sample. On the 4-2 plus Dex sample, however, the low levels of PAI1 compensated for the low levels of UPA and UPAR, ultimately leading to the degradation of the gel.
So in conclusion, we found that fibrin gels were able to support the proliferation and differentiation of DPSCs independently of Dex in vitro. Interestingly, fibrin had receptors for DPSCs to bind to and differentiate. We also found that gel dissolution times could be controlled by varying dex treatment and also fibrinogen concentration. Our data also indicated that on fibrin gels, dexamethasone actually inhibited differentiation of DPSCs by downregulating OCN, interfered with the DPSC's ability to sense substrate deformations, and slowed fibrinolysis by upregulating UPA and UPAR. From this, we can conclude that fibrin gels would be extremely effective scaffolds as they have controllable biodegradability and can independently induce differentiation of DPSCs.
In the future, we'd like to determine the specific receptors on fibrin that induce the differentiation of dental pulp stem cells. Future evaluations involving RT-PCR across multiple days will be needed to determine the differentiation lineage of the cells. Scanning electron microscope imaging will be needed to assess biomineralization for the formation of dentin-like structures, and further tests are necessary to determine the suitability of fibrin as a cell delivery vehicle for DPSCs in vivo. Fibrin also has promising future applications in 3D printing and cell encapsulation because thrombin is non-toxic and highly efficient. A sample process of 3D printing is shown in Figure 20, in which a syringe containing fibrinogen and DPSCs is injected and gelated into a support bath containing thrombin. We'd like to thank Stony Brook University and our mentors, Dr. Miriam Rafailovich and Dr. Marcia Simon, for all of their advisement and support. We'd also like to thank the Siemens Foundation, Discovery Education, and George Washington University for this amazing opportunity. Thank you for your time.
Thank you.